Purification is really a classic technique, century-old technique called chromatography. People use this for protein purification, especially in pharmaceutical area since last century, 70s, 80s. And for chromatography, what you do is you culture your bacteria or mammalians or whatever. You kill the cells and have a lysate mixture of many things go through a chemical support. Hopefully, the chemical support could distinguish your protein from other things. And in nature, cells doesn't do this. They don't have a sort of chromatography or column to purify their protein. For example, in our body, we producing insulin all the time, and they are purified, stored in beta cells.
Welcome to the Smart Biotech Scientist podcast. I'm your host, David Brühlmann, and today we are diving into the fascinating world of synthetic biology and AI-driven protein design with Haotian Guo, who is the CEO of Ailurus Bio. From creating the first synthetic membraneless organelles in bacteria to pioneering machine learning approaches in protein design, Haotian is revolutionizing how we think about protein purification and cellular programming. In part one of this conversation, we'll explore his unique perspective on bioprocess development and the journey that led him to found Ailurus Bio. Stay tuned. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Haotian, to the Smart Biotech Scientist podcast.
Yeah, well, thank you for inviting me. Really energetic opening. Okay.
Share something that you believe about bioprocess development that most people disagree with.
So there's one interesting topic I really wanted to share. I talked to many people who taught or still teaching bioprocessing in the school, universities, like this. One time I mentioned about the idea that, okay, we really should have all this bioprocessing like growing plants, right? You just put a seed and start to grow, automatically control everything by itself instead of right now, we have a huge tank and all the complex facility to control when we're going to add something, when we're going to feed more stock. People think, okay, it's just sci-fi. And I was thinking, hey, coming from all the agriculture, you have your fruit and crops all doing this way and not fermentator. But, well, I think that still technology is not there. And we probably going to see a very long time to really have the technology, have the whole ecosystem to adopt this.
Wow, fascinating. That's amazing. You have worn many hats in your career. You have been a PhD researcher. You have become now an entrepreneur, and along the way, you had some other stops. So can you draw us into your story and tell us your key learnings along the way?
Well, my personal story is really motivation driven. I think that it's much easier for me when I need to choose different options. My peers, they have their difficult time. Oh, I have an offer from big pharma company. At the same time, I could do postdocs in some great labs. What should I do? I usually don't have these troubles because in the very beginning, I know what I want to do, motivated by my scientific interest. And that's how I get into the synthetic biology, because I initially trained both in physics and biology. If you look into how people study physics, everything is well designed or properly controlled. It's completely not the case in biology. The closest form for that is synthetic biology 15 years ago. So that's why I jumped into this field. Soon later, learn about the iGEM, the competition, where people to compete about their SynBio projects and so on and so forth. Eventually, at the point when we start to do something about AI and synthetic biology combination together, I realized, man, these things could really move very fast. And it's very unlikely you could stay in academia and do this in a proper way to give the best impact to the society. That's when I jump out of the academia to the startup.
Did you plan to become an entrepreneur, or was more like by coincidence?
Good question. Well, I would say there is a sort of deterministic factor talking about, well, what I just mentioned, academia after centuries of evolution, the structure is very stable and rigid. And if you want to do something really wild and crazy, you're going to find alternative paths. I think the same story happened. Many other people like Demesis started DeepMind. It's exactly the same reason he wanted to do AI research, but there's no place in academia for him to do that. But there's also another thing I think is totally coincident. That the lab I'm in, although at the time all our professors, PIs, they have no interest in industrial or startups, but many people, previous students in the lab, really build this ecosystem in Paris for entrepreneurship really autonomously. I could give you a few examples. It's AI company for biology, especially in clinical trials. There's another company called Illegal Bioscience, one of the largest competitors for Illumina. MGI in China, the sort of tech lead is also from our lab. So many people study science before and then go to industry. Give me sort of a window to understand what it's like in the other side. So that also sort of a motivation and driven force for me to move towards a startup world later.
And as you moved from the first synthetic membraneless organelles in bacteria to now funding your company, Ailurus Bio, what was that key experiment or moment when you realized this technology could transform protein purification?
So there are two parts when we try to do this. So one is at the time I'm still in the lab. As a PhD student, we had very early on in 2017, we had the first demo for this synthetic organelle. Initially started as a sort of iGEM project. I recently had a blog talking about this story. We have several other projects in parallel in the lab where people need to purify proteins. And we train students, so complicated, and they cannot never learn and purify the protein in the proper way. One day my colleague said, okay, why not just, how can you do something to invent a new technology? Come on, and solve our problems. So that's sort of the motivation at the time. We try different designs to see how we could extract the protein out of the synthetic organelle. We didn't succeed at the time. So when we started Ailurus as a company later, still, we got the same problem. Our technicians, scientists need to purify protein, and they think, well, the traditional workflow sucks. And could you do something better, way better than this? And it's the second time I think about, okay, well, for the second time, I have more knowledge, more understanding, and deeper insight into how the technology we see in this synthetic organelle, because we did many experiments before. And this time we finalized methodology where we could really use this inducible self-cleavage to let the organelle to release the protein out of it automatically and then get super high purity and yield from this technology. Well, I would say this is really a technology invented for our own purpose because we need to do a lot of protein purification by ourselves. And when we launched it early this year as a toolkit, as a product, standalone product, that is similar process, passive way, where we deliver many customized projects, sort of like a CRO model for many customers. And some of them saying, hey, you transfer me a very weird strain producing protein so well, and I want to understand how you made it and do it to my other projects. When we see many of these common requests, we think, hey, maybe just have the toolkit as a reagent kit to all the customers. They probably could use it in-house without telling us what the protein they are using or studying. So I would say really we learned a lot during this process. As a first-time founder, many things could go completely different direction as you expected at the beginning. Yeah.
Can we zoom into a bit more how the technology works? Because you're using bioprogramming, you're using organelles to purify, but can you tell us maybe from a high level, like how does the technology work?
Yeah, so for people who are listening to the podcast not familiar with purification, purification is really a classic technique, century-old technique called chromatography. People use this for protein purification, especially in pharmaceutical area since last century, 70s, 80s. And for chromatography, what you do is you culture your bacteria or mammalians or whatever, you kill the cells and have a lysate mixture of many things go through a chemical support. Hopefully, the chemical support could distinguish your protein from other things. And in nature, cells doesn't do this. They don't have a sort of chromatography or column to purify their protein. For example, in our body, we producing insulin all the time, and they are purified, stored in beta cells all the time. How they do it is using organelle to sort the protein into targeted location and release them when it's needed. And synthetic biologists try to mimic synthetic organelle structure for decades. We have failed so many times. And until very recently, there's a new class of organelle, not the membrane-bound organelle that we are familiar with, like mitochondria, but rather it's a membraneless organelle. They do not have a membrane, but they assemble a distinct structure using phase separation, liquid phase separation. Just like when you mix oil and water, they will separate. In this way, we could build a synthetic organelle in bacteria or many other organisms, and we could program our target protein with a tag to go into the organelle. And of course, we could induce them to release a protein from the organelle. In our specific case, we use a self-cleavage, very similar to how natural organelle process a protein. Usually, we have a signal sequence in the N-terminal of the protein. When they enter the organelle, it will be cleaved to go for the next destination. So in this way, the whole workflow is very simple. You just grow your cell. Then you lyse the cell to remove all the impurity, the whole cell components. And in this way, the organelle, which is 1,000 times or even 10,000 times bigger than your impurity, will stay in the pellet in the bottom of your tube. Then you add a new inducer to release all the protein. All your protein will come into the supernatant where right now you have no other proteins or impurity in that solution. In this way, we could really get a high yield, high purity, very simple workflow.
So in the body, obviously, we're talking about small scale, or shall I say very small scale. Now, if we talk about the industry, probably the scale will a bit bigger. So what scale are we talking? Is this milliliters, or could it go up to liters or even more? Or how do you envision this technology?
So right now, it's quite stable for us to scaling down, of course, in the lab. That's easy to do. Well, at the maximum, we have done 1 liter, 2 liter in bioreactors, small ones, the lab scale small one bioreactors. And it could really go downscale, 10 microliter, 100 microliter, that you could really use very small microplates to high throughput experiments. Well, of course, another direction more relevant to industry is how to scale it up. Right now, we are doing some pilot experiment to try to scale it up to 50 liter to 100 liter. Then it will become more relevant to the industrial production. But as why people don't buy in my fruit theory to just grow everything automatically is when you really have a bio sort of program process in your microbe, it's not their nature function. When they grow very, very large, many, many generations, they probably will lose your plasmid, mutate the system, and do something else, right? So we will see how good it will go for scaling up. Definitely, it will require lots of optimization, particularly probably need to tailor for specific proteins, I would say.
And what is the purities you're seeing? Because most of the people are familiar, let's say, with protein A, for instance, for a traditional antibody or fusion antibody purification. Is that similar? Is it very different? Or how does this play out?
Well, yeah. So right now, I could share a little bit of technical details into this. Right now, we figured out, okay, so purity actually really, unlike the traditional workflow, the purity depends on how accurate you could capture your protein. For example, protein A could capture something else, not the full-length antibody. well, his tag is more common. Any protein with enough histidine could bind the colon. But in our case, the purity could be ensured by these organelles. They are very specific because everything is programmed by biomolecular interactions. We could easily screen for interactions very specific. Right now, we could see under the microscope exclude any protein that are not specific to bind to the organelle. In the future, we could even enhance this recruitment. Where the impurity coming from is actually from the whole cell. If the cell is not lysed completely, and if it's not washed properly, then we'll see a lot of impurity in the downstream processes. This is what we try to do. For example, in the lab scale, people do sonication. That could really disrupt the cell completely, very, very nice. Then we'll have very high purity up to 98%. In another way, if you do high throughput, you cannot just disrupt them one by one with your needle. Then you need a lysis with a reagent, with a buffer, where at that time, some of the cell may not be lysed in the previous step, and then after release some impurity, the purity will go down to roughly about 80% or 90%. But luckily, many people who do high throughput screening, they don't care about impurity at all. Some people even still doing cell lysis for their activity assay, for example. So right now, we are hitting the point where most of the customers will be happy about the purity level.
When I come to you with a new protein, to what extent do you need to do new computing, like developing your organelle from scratch? Or do you have some libraries, or how specific are these organelles then to the protein?
Yeah, so right now we really apply a modular approach, which means for any protein, we have exactly the same organelle. So basically, when we sell a kit, it's the same organelle, right? Of course, there's space for optimization. For example, some organelle may have their electrostatic properties better for one kind of protein, not for another. In that sense, we do have a library internally to screen for better conditions. But right now, we ship one is based on the RNA molecule, also the same as what we published earlier in the Cell paper, that has the best property for all the protein expression and purification for most of the protein in our hand. And how protein will be specifically recruited is really to adding a tag right now specifically targeting the RNA. So basically, in our kit right now, we have one plasmid encoding the RNA organelle, another plasmid for expression vector. You just need to clone your protein into that vector. We plan to see other options. For example, many people still doing histap purification in the lab. They probably really do not want to do extra cloning, kind of troublesome for many people. And we probably will launch new organelles specifically optimized for those proteins. When you already have a histap, you just add a new plasmid to express the organelle to go through all the processes. That would be nicer for those kind of people.
And that wraps up part 1 of our conversation with Haotian Guo. We've covered some groundbreaking insights into synthetic biology and the future of protein purification. Join us next time for part 2, where we will dive deeper into the technical aspects of synthetic organelles and AI applications in biotech. Before we go, if you're struggling with biologics development and looking to fast-track your process, I can help. As a fractional CTO, I provide on-demand expert guidance and strategic insights to help biotech companies accelerate their development timeline without costly delays. Visit BrühlmannConsulting.com to book a free consultation call. Let's transform your biologics development journey together. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.